What's up guys, welcome back to the channel, and today we're going to start a new playlist, something I wanted to do for a while, and we're going to be talking about one of the most important topics of biology when it comes up to the IMAT, that is bioenergetics. Right in the last edition of the IMAT, you're going to see so many questions of this topic that you're going to start asking yourself, what the heck are these guys doing? But it's no surprise, 2024 was just like that, and it kind of seems that now that's the pattern. So this is the first video I'm going to be recording. The idea is to cover all the steps of bioenergetics, so I believe this playlist might have 4 or 5 videos until we're good. Last thing I want to mention before we jump into the video is that I organized myself finally, and we're going to have videos every single week. So if you're new to the channel, you're not subscribed yet, don't forget to hit that subscribe button and give it a like if you enjoy this content. Let's get into it. Alright gang, so here we are, bioenergetics. What are we talking about? So we're going to be talking about all the different processes that our body undergo in order to produce energy. This is the main goal, and this is the main topic of our video. Of course, the most important of them, the one that is most requested in exams, is cell respiration. This is a topic that is considered by a lot of people one of the most complicated topics in biology, but the idea of this video is to talk about all these in a way that is understandable, go step by step, and in a way that you can retain this knowledge in order to mobilize whenever you are requested. So in this first video, we're going to be talking about glycogenesis. Further on, we will be talking about Krebs cycle and then oxidative phosphorylation. So we can just climb this stairs step by step. And later on, I will be leaving some videos just to talk about some actual process that our body undergo to produce energy, such as fermentation, such as glycogenesis, such as other processes that could be also asked in the IMAT exam. If you have any questions or suggestions, just leave in the comments and I'm going to be more than happy to help. Let's move on. All right, so there's no other way to start talking about biochemistry without talking about the ATP. This is one of the most important molecules in our body and is responsible for almost every single reaction that our body undergo. The ATP it's basically considered the energy currency. Not just that, it's also considered a universal energy currency. And the reason is because every single being on this planet requires and depends on this molecule. There's no single being that can survive without ATP. So let's take a look at the molecule itself. This is something that it really looks like IMAT question, understanding the little bits that is part of the whole molecule. Something that is important to mention is that this molecule is really high energized. As you can see here, we have 3 molecules of phosphate. That's no surprise. The T here in ATP stands for tri, stands for 3. Okay, 3 what? Phosphates. Other than that, we're going to have a sugar here forming the base of this molecule that is going to be a ribose. Okay, this might be something important to remember, like what is the sugar that is part of ATP molecule. And we're gonna have a nitrogen base here that is going to be an adenine. So it's basically almost like a nucleotide. Okay, consider that a nucleotide only has 1 phosphate. If you just cut this part off, you have an adenine nucleotide, the same one that is part of your DNA. Basically, this is a molecule that is so high energized, it's almost like a spring ready to pop. For that reason, it's used for so many chemical reactions. So when you think about chemical reactions, there are chemical reactions that they happen easily. Okay, they're ready to go. Think about like petrol, okay, in a motor of a car. The explosion is something natural. It's just gonna happen. You know, like the small little amount of energy, it's enough to get that reaction going. That's why it's so dangerous. But on the other hand, there are other reactions that they don't go really easy. So you really need to make a lot of effort to make that happen. So whenever we have a crack in this chain here and we have 1 phosphate just breaking out, that's going to be releasing so much energy that it's going to impact on the molecules around. Another very important subject you're going to have to keep in mind, not only for biology, but also for chemistry, is the redox reaction. The redox reaction is a class of reaction, and put it in a very simple way, it is the reactions where we're going to have electrons being transferred. This is really important for chemistry, by the way. There's a lot of questions about this. I want to cover this in another video, but let's focus on biology. So it's a reaction that happens in pairs. For electrons to be transferred, we're going to have someone that loses and someone that receives it. And this is really important for biology because when you think about you're eating something, okay, you're putting nutrients in your body, carbohydrates, they're large molecules. The first thing that's going to happen is that we're going to break this down to small pieces. It's going to start digesting in our mouth. It's going to finish in our intestine, stomach, and then we're going to have just these molecules of glucose. But we still need to break these down, okay? And then the responsible one to do that, it's going to be the cells itself. So the glycolysis, that is the first step of this reaction. It's a process that happens in the cytosol. We're going to talk about this more specifically. But the thing is, we're going to be breaking this down and we're going to be removing electrons from them. The molecule of glycolysis, it's going to undergo a lot of oxidations. The oxidation, you can't forget this, is the one that is going to be losing electrons. On the other hand, the one that's going to receive electrons is going to be the reductors. For now, just keep in mind that this is just going to be so we can transfer these electrons to be used in order to produce what? ATP. This is just a little spoiler, okay, of an example where we're going to have a redox reaction. We're going to have these little guys, files that we're going to call FAD, and they're going to be very important because they're going to be transporting this electron. Right now here we have somebody oxidizing, somebody reducing. Can you identify it? I'm kind of feeling like Dora Adventure right now. Can you identify who is reducing? but serious. Basically, you don't see here right now, but it's going to become clear for you. This little guy here, he's just ripping out 2H2, okay? So it's going to have a hydrogen that is going to be carrying electrons, not only the hydrogen. So if he's carrying electrons, he is reducing, and the other intermediate guy here, he is oxidized. We have a question from 2023. Let's just jump into redox reaction plays a vital role in many cellular processes. Which of the following statements and examples best describe redox reaction in living things? Redox reactions involve the transfer of electrons where the molecules that gains electron reduce and the molecule that loses electrons is oxidized. Perfect. This is a definition of a redox reaction. You're going to see that the other options here doesn't really make that much sense. One thing I want to talk about the IMAC questions is that there's questions that they just kind of scream into you the right answer because the other ones doesn't even make sense. Let's get one here, for example. A redox reaction involves the observation of light where the molecule that gains light is reduced. There's nothing even close to it. So these questions here, if you have like a little understanding of the topic, you're gonna find the answer with common sense. So that's good. Moving along, I gave you a little spoiler about the FAD+, but I didn't tell you that he has a little friend that is called NAD+. These two little fellows are gonna be really important because they're gonna be electron carriers. First thing you have to think about is where are they coming from? I mentioned that they're gonna be coming from the nutrients, everything that we eat. More specifically, the carbohydrates, okay? If they're coming from there, these little guys that are losing the electrons are going to be oxidized, all right? On the other hand, the NAD and the FAD will be reduced. Now, there's two different forms we can find these elements in our body. It is by the oxidized form. That's why we have a little plus here. Remember from chemistry, whenever we have a plus, a ion that has a plus, we're gonna have a cation. I don't want to bring any PTSD memories here, but this can help maybe to remember. And on the other hand, we're gonna have the NADH. That's the form that is reduced. It's carrying the electrons already. So we're gonna be talking about these guys a lot in this video and in the further video as well. So I don't want to waste too much time talking about them right now. The only thing I want to mention is about the nature of these molecules. These are considered enzyme cofactor. So they're not enzymes itself, but they are very important helpers of the enzyme. If you remember, an enzyme is going to be a very important element to help a reaction to happen, accelerating the process by minimizing the energy required to make it happen. And most importantly, the enzyme will not change at the end of the reaction. It's going to remain the same as it was in the beginning. But these guys here, they're going to be important because they're going to help out the enzyme to break down those molecules. So they're going to be receiving the electrons that those molecules are going to release. And they're going to bring this all the way to the mitochondria to produce the ATP. Now we're going to see this in details, but just keep in mind that there are enzyme cofactors. They're not enzymes. Now, talking more specifically about the cellular respiration, okay, finally. The first step that we're going to be talking more deeply is going to be the glycolysis, but just a overview here. Just want to frame that there's a difference between the systemic respiration and the cellular respiration. The first one is the one we usually breathe in and breathe out. Okay, this is the systemic, and the cellular respiration is going to be the process that our cells use to produce energy, ton loads of energy, and they do that by binding the glycose, okay, the carbohydrate glycose, with oxygen. So in some way they're kind of connected, right? And the byproduct we're going to have here is going to be CO2, water, and most importantly energy. Something maybe interesting to mention is that the oxygen from water here is not coming from the carbohydrate but from the oxygen here. All right, I know this could be intuitive, but the oxygen from the CO2 does not come from here but from the carbohydrate. Now another thing I want to mention, I know the IMAT does not ask a lot about photosynthesis, but it's good to have the big picture here. Okay, so the carbohydrate that our mitochondria, our cells, uses to produce energy, it comes from the plants at the end of the day. Okay, because this little guy here, chloroplast, it's organelle, if you remember, it's important organelle from plants, and it uses CO2 to produce glucose. So it does exactly the opposite reaction. Now, something that is important as well when talking about cell respiration in a generic way is understanding all the steps and also the location of these steps. All right, so we usually split cellular respiration into 3 steps or 4, depending. This is the order we're going to find. Okay, so first we're going to have the glycolysis, the one we're going to talk about it today, and it takes place in the cytosol. On the other hand, we're going to have the pyruvate oxidation. That is an important process. We're also going to talk about this, and it takes place in the mitochondrial matrix. That is basically the inner side of this organelle here, such as the Krebs cycle, the one that usually people struggle a lot, that has a lot of names. Finally, we're going to have the oxidative phosphorylation, right? This one here is going to be the responsible one to produce those ton loads of ATP, all right? And they're going to take place in the inner side of the mitochondrial membrane. Remember that this organelle has 2 membranes, the outer one and the inner one. It's kind of related to that endosymbiosis theory that is very interesting. So digging into glycolysis, as we mentioned, it's going to take place in the cytosol, and it does not require oxygen to happen, okay? And this is going to be important as we're going to see in other processes such as fermentation. So this first step here of cellular respiration is going to happen outside of the mitochondria. It has nothing to do with it. In this other slide here, we're going to have an overview of the glycolysis, what happens at the beginning and how does it end. We're going to see this with more details, but basically we're going to come from a glucose molecule that has 6 carbons, and we're going to end up in a different molecule called pyruvate that has 3 molecules. In between, we're going to have some ATP being produced, not a lot, but something just to get going. And also we're going to have some electrons being transported. So this little guy here, NAD, remember him? He's going to be carrying some electrons that are going to be important for our last step. Now with a little bit more details, if you notice, we're going to have ATP being consumed. Okay, we're going to need ATP in the first part of glycolysis. But at the end of the day, we're going to have these ATP back, not only the 2 we used, but also some extra. So that's why we usually separate glycolysis into these 2 steps. The first one is going to be the investment step. We're going to invest literally ATP to gain money. We have to lose money. And then we're going to have the payoff step. That is basically the step where we're going to get back these ATP and also produce some NADH. All right, so this next slide here shows the glycolysis process with a lot more details. I know it can be a little bit overwhelming with all these enzymes, these arrows, these names, but don't worry. It's gonna get worse. No, just kidding, but seriously, I don't want you to freak out and try to memorize every single thing. What I'm gonna try to do here right now is highlight the things that the IMAT have already asked and the thing that I believe the IMAT could ask, because like true being said here, the IMAT is getting a lot more specific when it comes to bioenergetics, and they're kind of forgetting about the other steps, the other things of biology that they mentioned. And I believe that this is kind of to prepare you for what's coming in the following years, like chemistry and biochemistry. So you don't have to worry, you're gonna have a lot of fun with all these names. And of course, university, you're going to deep yourself in much more deep waters. Okay, so just recapping, the glycolysis is this energy process that happens in the cytosol. And it's going to use the glycolysis to start this pathway. So how do we get glycolysis? Through our diet. Okay, we can eat a lot of complex carbohydrates. They're going to be break down into different monomers. Okay, we're going to have glycolysis, fructose, galactose, but primary, we're going to use the glucose. Okay, later on, the fructose and other carbohydrates are going to be transformed in a way that can be used, but the glucose here is the star of our show. So as you remember, it's going to start being break down in our mouth through our saliva and enzymes, and then it's going to end up on our stomach, intestine, and also everything that we couldn't absorb in our digestion tract is going to be finished in our kidneys. Once it's all in, it's going to be flowing in our vessels, and then it's going to be absorbed in our cells. Now, glucose can be easily absorbed in our cells whenever it's required. We have receptors, proteins in the membrane that are going to be responsible for doing that. Okay, more specifically, we're going to have different type of GLUTs. These are the proteins that are going to transport the glucose. That's going to happen through necessity, okay, through a difference of gradient. So if you have less glycose inside of the cell and more outside, it's going to passively enter the cell. The other way around could happen as well. So it's a process that doesn't require ATP. If we have less glucose outside and more inside, it's going to naturally just walk out of the cell. And the cell does not want that. So the first step of the glycolysis is making sure that glucose is not hitting out our cells, okay? In order to do that, it needs to kind of change a little bit the glucose structure. So we're going to have an enzyme playing an important role here that is called hexokinase that is going to add a phosphate to the glucose molecule. Doing that, it cannot access the cell anymore by those transports because it doesn't recognize it. So that's a really smart strategy for our body, guaranteeing that glucose is going to stick into the cell. It might be interesting to remember the enzyme that does that, the hexokinase. That's the most important one. We have another one that is called glucokinase. Now, that's only going to happen in the liver. This much of detail, I don't think it's important, but I just want you to realize that every time we have an enzyme that ends with kinase, it's already telling you what's going to happen. It's going to add a phosphate. Remember, we're going to have ATP being used at this stage. So we're going to use one ATP molecule in order to pass on that phosphate to the glucose. Once that happens, we're going to have the second step of our glycolysis that is going to be a isomerase. So we're going to have an enzyme called phosphoglucose isomerase that the main role is just to change a little bit and rearrange the elements. So remember, an isomerase is responsible for changing the position of certain molecules, okay? There's a proper reason for that, but we don't need to dig ourselves into it. But just notice that instead of a glucose 6-phosphate, we're going to have a fructose 6-phosphate. Now, after this step, we're going to have another phosphorylation, and for that, we're going to use an enzyme called phosphofructokinase. Now, notice that leads us to a substrate called fructose 1,6-diphosphate because it has 2 phosphate in that molecule. Now, from these 3 enzymes, I think the most important one to remember is the hexokinase, and maybe just keep in mind the phosphoglucose isomerase because it's kind of a bridge knowledge. Understanding the isomerase concept from chemistry here in biology might be interesting. Now, moving on, we're going to finally break this substrate into 2 to kind of start getting close to our final molecule that is the pyruvate. And the enzyme responsible to do that is going to be the aldolase. It's not going to be a symmetric break, you're gonna have the formation of two different elements that they can interchange between them. But the one that is gonna be responsible to move on in our glycolysis process is gonna be the glycerate aldehyde 3-phosphate. Now, at this point here, I don't want you to memorize any substrate, any enzyme name, because this is a lot specific for the IMAT, and I don't think they're gonna concentrate into that detail. But just understand a little bit of the process, because from now on, what's gonna happen here, we're gonna have the formation of a NADH. So a cofactor is gonna play an important role of carrying some electrons. And remember, this is gonna happen twice, because once the molecule is broken into two, the other one is going to be converted to the substrate here, and then we're gonna have once again some electrons being transported through these cofactors. After that, we're gonna finally have the formation of the ATP. Remember, from now on, we're talking about the payoff phase. So the ATP is gonna start being synthesized. And once again, we have a kinase here playing this important role of passing on the phosphate. And just before we form a molecule of pyruvate, we're gonna have a phosphorylation happening here. Remember the pyruvate kinase, because this is the one responsible to form the pyruvate and also to create another molecule of ATP. So we have two molecules of ATP being produced here with just half of a molecule of glucose. So at the end of the day, we're gonna have 4 ATP being synthesized. Once again, I know it can be a little bit overwhelmed, but there's two different approaches you can give to this. First one is a little bit more generic, just remember the most important things. And the second one is deeping yourself in. The first one, what is really important for you to remember is that we're gonna split glycolysis into two. The first phase is gonna be the investment phase, and the second one is going to be the payoff. So in the investment phase, we're going to have to use 2 ATPs so that in the payoff phase, we can produce 4 ATPs. We're also going to have the formation of 2 NADH, those electron carriers. Just remember, we're going to have one here, but this is going to be doubled. And most importantly, that the glycolysis take place in the cytosol. And the second approach is going to be by remembering the name of some enzymes that might be important for the questions. The first one I want you to take in mind is that the exokinase is going to be the first enzyme, and it's going to add a phosphate. Other than that, the isomerase is going to be responsible for changing the place of some atoms. And also the aldolase, that might be important because this is the enzyme that is going to start the formation of pyruvate. It's going to be the one that's going to break these two molecules into two. Just remember, they're not going to be symmetric, okay? So we're going to have the formation of two different molecules. And the last one that I think it might be important to remember is going to be the pyruvate kinase. Now remember, every time we have a kinase, there's a phosphate playing a role here, okay? So in the investment phase here, all the kinases are going to add a phosphate to the substrate. And on the payoff phase, all the kinases here are going to be responsible for removing a phosphate from these molecules. That's how we're going to produce the ATP. So just keep this pyruvate kinase into your heart, and remember that's going to be the last one, and it's going to form the pyruvate. And this slide here just synthesizes what we just talked, and it's usually common to bring the results of the glycose as this way. We're going to have the formation of 2 pyruvates. We're going to have the net gain of ATPs as being the number 2, and we're going to produce 2 NADH. Always 2, 2, 2. With this in mind, you're good to go. Now, before we wrap up this video, I'm just going to bring some questions that the IMAT organized. At the end of this playlist, I'm planning to record a video just with questions of bioenergetics. So leave a comment if you think this idea is good. 2026, which of the following are respiratory enzymes? So remember, enzymes usually end with A's. Looking at the options here, we don't see anything related, but we see a FAD and an NAD. We know that these are not enzymes, but these are cofactors. Any options that have 2 or 3, we can just eliminate it. So A, B, C, and E. So we don't even have to worry about the acetyl coenzyme A. By doing this, we can eliminate all the other options, and that leads us to the letter D. And it makes sense because this is not an enzyme, as we're going to see in the following videos. Now, this last question here, I'm not going to answer. I'm going to leave it to answer in the next video. If you guys have an idea of the correct option, leave in the comments and tell me why. And in the next video, we're going to be talking about the fate of the pyruvate. So we're going to be covering the fermentations, the most important things about it, and we're going to talk about the next step in the cellular respiration. So guys, if you enjoyed this content, don't forget to like the video. Also, if you have any suggestion or question, just leave it a comment, and I'll be seeing you guys in the next one.